yeah, which is no longer increasing rapidly. So normally I would say that Simon Segar doesn't need introduction, but I think actually he does to some extent, because there are so many new people at the department who have come there since he left. So happy to have Simon here. This is his first, I think, first Kokomo since he left for the happier shores, and he came to our department as a postdoc with the aim or task to introduce phylogenetical perspective to our ecological studies of food webs, which he duly did, but then as a sort of unexpected extra also introduced the chemical secondary metabolite analysis in the plant insect food webs. And so that work still continues with quite intense collaboration, especially with Martin Wolf's and mine laboratories. But you know, for those who are new, it's good to know that Simon is active in these two fields, and he's now at Harper Adams University, as you can see, and actually also to my surprise is coming with a completely different topic. So this will be definitely new for all of us, no matter how long we have been at the department. So again, thanks for joining, and the seminar is yours, Simon.
Okay, thank you, Wojciech. Thank you very much for the introduction. So yeah, I'm very happy to be able to present some ideas basically at the seminar. Unfortunately, I've got contracted COVID in the last week, which is basically given me a whole range of different symptoms. Today seems to be breathlessness, but and a bit of a cough, but we'll see how it goes. So as Wojciech said, I started to work in the UK in 2018. So I moved to Harper Adams, which is a smallish university in the middle of England. So it's set in a very nice location in Shropshire. And it's got a history of agricultural work. So I'm trying to conduct a line of research that's both interesting from a pure perspective, but that's also kind of got some applied benefits that's going to help people in more immediate term as well. So this is all entirely introductory. So we do have one MSc student, one Kenyan student starting soon on a Harper funded project. That will start hopefully in the next couple of weeks. We did have a PhD student, but unfortunately she had to stop her studies due to health reasons. So we don't have a PhD student anymore, but we are advertising. So we'll be advertising for another position soon. So please keep an eye out on that. And we've got a couple of we had one unsuccessful but well scored NERC bid recently, and we've got another NERC bid for a developmental grant in. So this is all very much kind of ongoing. The collaborators in Kenya are Esther Kioko at National Museums Kenya and Mark Atenyo at University of Embu. So these are both very good ecologists, entomologists, and we've been really lucky to be able to work with them. So I do have a kind of grand plan, which I'll come back to at the end. And this is how my first grant proposal kind of was structured. It got kind of cut down to size eventually, but it's interesting to see that not many people know that there are quite a few species of coffea, so coffee, that grow in the wild. So it's not just your arabica. So if you can see my mouse, which you should be able to, you can see there's a clade here. This is a phylogenetic tree. So of course, I'm still trying to use a phylogenetic context that Wojciech suggested. You can see there's a clade here, and the values on this tree are basically a trait value, and that trait value is the caffeine content in green beans. So these are the coffees that we drink. So there's Canaphora or Robusta. Arabica is not actually in the tree, interestingly enough, and I'll tell you why very soon. And then these are all Malagasy clades. So you've got your high caffeine West African, Cameroonian, Congo species of Brevipes, Canaphora, Congensis humilis liberica. Liberica is actually a third coffee species that people use for a rootstock. Stenophylla. Stenophylla is interesting. We'll talk about that in a minute. Then you've got your East African group, Arabica, Eugenioides, etc. And then there are over the bulk of the, well, not quite, but almost half of the genus is in Madagascar, where you have typically very low caffeine species. So there's 2 species in Madagascar that have moderate levels of low to moderate levels of caffeine. The caffeinated species are typically west to east African, and really there's a kind of almost caffeine gradient as you go from west to east. So there's an idea that there's lots more pressure in terms of herbivory. In West Africa, it's wetter, more humid, more tropical forest, lowland forest, and there's stronger herbivore pressure, and you've got this selects for stronger defenses. So it's kind of classic argument really for that chemical ecologists like to employ to explain the distribution of chemical diversity. So it makes a nice system because it's chemically diverse. There's not just caffeine in there. I'll tell you what else is in there. People rely on it, so it's a $100 billion industry, and it's probably more important than many other industries because lots of people who are subsistence farmers rely on it. So the kinds of people that wouldn't have work otherwise are able to grow coffee and sell. It's a luxury product, the largest grossing legal narcotic. Certainly the largest grossing legal alkaloid, probably nicotine's chasing its heels a little bit. But it's under pressure. So we're facing pressure from climate change. Some of these species are growing, reducing in their range. Some of their pest species are moving around as well, which I'll talk about. And it makes an interesting system for studying chemical diversity and how chemical diversity is generated. So whether it's due to herbivory pressure, whether it's due to the environment, some combination of the two. And what's arabica? So arabica wasn't actually in that phylogeny. So arabica is a polyploid. Actually, it's a cross between Eugenioides, and this is Eugenioides here, a West Kenyan species. So it grows on the border between Kenya and Uganda. So a study system which lives somewhere interesting, with the hope of eventually visiting once kind of COVID's over. And it's a hybrid basically between Eugenioides, a wild species, and Canaphora, which is robusta. And these wild coffee species make a tremendous resource. You can see there's lots of them. We only really drink 2 or 3 of them. They've got different caffeine content, and they're potentially a really important resource for understanding chemistry. So as well as the purine alkaloids, caffeine and triglycerine in coffee, you've got various polyphenols, chlorogenic acid, caffeoleoquinic acids. These are the acids that give coffee that bitter taste, but likely have some defensive function. So chemically speaking, coffee is quite interesting, and it has one of these chemicals which is extremely valuable. As I kind of hinted at, it's under threat from climate change in various ways. So there's been some really interesting research. None of this is mine, but it doesn't make it any more relevant for a research seminar. This is the coffee berry borer. So that's a really serious pest of coffee. It bores in the berries, as the name suggests, and can destroy whole crops. There's a really nice paper in 2011 in PLOS by Jamilo et al. And they basically showed that under current climate change models, we're going to see the coffee berry borer expanding its range quite considerably. So we can expect these pests of coffee to get worse as we go through time. There's also a huge cost to these existing species in terms of where things can grow. So Kenya's becoming drier. Large parts of the range are becoming drier, hotter. So where we could grow coffee 20 years ago is not necessarily where we can grow coffee nowadays. So there is even so Stenophylla, that's the reason I said it was interesting. This species here, Stenophylla, you can see it's fairly restricted to West Africa. This was rediscovered recently by Aaron Davis, who's kind of helped us with some of these proposals. Tastes a bit like arabica, but it's got a slightly higher climate envelope, so it can persist a couple of degrees hotter than arabica. So we're trying to mitigate some of these costs on the 2 or 3 cultivated species by looking for alternatives in the in the wild population. So that's also genes for defensive traits, genes for coffee production, caffeine production, taste, bitterness, Unfortunately, Eugenoides apparently tastes a bit like mud, so it's probably not a great candidate. But there are other coffees out there that do taste reasonably nice. I think Eugenoides probably doesn't taste too bad. It's compared to arabica and Canephora, it's not that nice. So coffee's under threat. It's an important industry and it's chemically interesting. So you can kind of see why it makes a good study system. So to complete the kind of trilogy of interesting things about coffee, it's also host to lots of pests. So it's Rubiaceae. This is a huge family of flowering plants, including things like Psychotria, which has got some more interesting alkaloids in it. And it's got a lot of insects associated with it. So the coffee pests are things like the coffee berry borer. So here we go, it's a tiny little beetle. Coffee leaf miner, also quite small and hard to get hold of photos of. And then the coffee stem borer. So we've got some beetles, the Lepidoptera, that are pests, but we've also got lots of other insects that eat coffee, wild pest species. And we're not sure, actually, we've got no idea, really, apart from a couple of studies by Bob Copeland on coffee beans, what eats wild coffee. So the overlap between caffeinated, so domesticated species or those that are grown in plantations, we really don't know what comes from the forest and eats these trees and what does it the other way around. So what kind of typical coffee pests and how they spill over into endemic or native forests. And remember that coffee's from Africa, but it's also grown around the world. So there's some really nice opportunities for studying coffee pests within the range, within its native range, when it expands its range in different situations and different climates. So it's a really nice system, and there's just so much to be interested in when you start looking into it. It's also obviously a flowering plant that's pollinated by a whole range of insects, so bees, wasps, large flies, butterflies, but it's really mostly bees. So outside of its native range, it's Apis mellifera, the European honeybee, that really does most of the work. And interestingly, there's kind of quite a lot of evidence to suggest that actually these social bees get addicted to the caffeine. So what happens is a bee will visit a caffeinated flower, and it will enjoy the coffee. It will be stimulating for that bee, and it reinforces the association of that bee between that particular flower, and then which becomes associated with the scent of the flower, the shape of the flower, and it will visit coffee to the point of exhaustion. So it will actually pollinate coffee until it becomes exhausted and can't make its way back to the hive. So caffeine is a really important, a big deal for social insects, because if they do get back to the hive, they'll also communicate where that caffeine is. So it's a hugely important pollination resource as well as a defense system. You also have things like carpenter bees and solitary bees that also pollinate coffee. So it's got an interesting network of pollinators and parasites and herbivores, which makes it nice actually, because you can then test for antagonism and trade-offs between traits related to the floral traits and leaf traits. So the first aim, and as I said, this is really very basic stuff. I'm not at a position, well, I am at a position where I wrote an incredibly complicated grant and was basically encouraged to scale it back a tiny bit. So right now, we're trying to just answer some of these really basic things associated with coffee chemistry. So we've got two projects going on. We've got Dev Cafe, which is a project that's been funded. No, that's been that I've applied for money for. And this is really a developmental grant that will allow us to hopefully cement those relationships with Kenya. So we can go to Kenya, they can come and visit us, and we can collect some samples in the wild, and we can do some basic chemical analysis. So the idea of this Dev Cafe project is that we can quantify the chemical diversity of 3 wild and 1 domesticated species. So it's pretty basic. What we don't want to do is just collect baseline data on plant chemistry. But later, once we start to study environmental factors, insect diversity, all these other sources of data, can we start to be a bit more predictive in the way that we understand these ecological networks associated with coffee? Another aim is that we also want to understand the Coffee and Cigarettes was a Jim Jarmusch kind of series of short films with Bill Murray in it. So I thought, because I didn't really have any data, either show other people's data or kind of make these obscure references. This is a study by Adler et al. In 2012, and this was in Nicotiana. So there is a link there that's coffee and cigarettes. And Nicotiana is interesting, and this study shows why. So basically, another phylogeny, another set of traits. You've got pollinator reliance in blue. So the ones with the big blue circles are heavily reliant on pollinators. So the others, you have some which cross or intermediate between selfing and outcrossing. And then you've got leaf nicotine concentration. And you can see that leaf nicotine concentration is generally pretty low in these species which rely on pollinators. So it's quite hard actually, and because there's some genetic correlations there. If you've got high leaf nicotine, you've generally got high flower nicotine. If you've got high flower nicotine, you put off your pollinators, and so you don't want to be well defended if you're trying to attract pollinators. So there's a really interesting antagonism between what plants do to attract insects and what they do to deter insects, and how these two selective pressures can interact to shape the chemical diversity or the chemical content of that plant at the end of the day. So I think one of the important things to think is to kind of understand, I guess, is that plants aren't just eaten by herbivorous insects. There's a whole load of other things that add selective pressure onto those plants. So we've got things like pollinators, microbes, fungi, parasitoids, all sorts of other insects and environmental factors as well that shape that overall chemical diversity in these plants. And another thing we want to do with this Dev Cafe project, what we've asked to get money to do is to understand the chemical, basically test the hypothesis that this chemical composition will vary among populations within species. So this is it's about as basic as it gets in terms of scientific hypotheses. Is there variation among and between populations within a species? And does this relate to the basic environmental conditions at each site? So if it's drier, it's hotter, do we see a different investment in chemical defenses? Obviously, this is just a fraction of the story. It's a small grant, but then we can start to link it to insects if we collect entomological data from the same plants at the same sites, and we can be a bit more sophisticated and do some more interesting research. We're also going to try, if we get funded, to assess the chemical composition of both flowers and leaves to see if chemical composition in leaf and flower tissues shape each other. So does leaf chemistry affect flower traits, basically? And that's the case in Nicotiana. So the correlation is different. In Nicotiana, you've actually got a kind of a negative correlation where you've got so lower leaf caffeine, lower leaf nicotine equals lower flower nicotine. In coffee, would expect probably the opposite. Would expect that there'd be high caffeine in leaves would equal high caffeine in flowers, which I realise is the same correlation when it's at the other end of the scale. I'm really not particularly with it at the moment with coronavirus, to be honest, but we'll get through it. So the second project that we've actually had funded, this is just a small amount of money, so it's just £5,000 from Harper Adams University. It's kind of pump priming money. It's not a huge amount of money, but with that, what we want to do, basically employ a Kenyan master student, so someone who will have done most of the taught part of their degrees, get them to start surveying insects and pollinators on wild coffee species. So we're gonna study a couple of wild coffee species and one domesticated species. Test the hypothesis that these insect herbivores will be specialized, because we expect these floral cues to be more species specific than, for example, the leaf traits. So we expect the herbivores to be specialized because we expect the chemical use to be more conserved, whereas the pollinators will be less specific, because we do see hybridization in coffee. So I realize I kind of messed that up a little bit when I was trying to say it. Again, the same excuse. We do see hybridization between species of coffee. So we see it throughout West Africa, we see it in East Africa. So there's a reason to believe actually that these pollinators aren't that specific, and that's why maybe hybridization is reasonably common in Coffea. The insect survey data will help to identify important pollinators. So also one of the species that we want to study is a CITES listed species. It's got about 30 individuals in one part of Kenya in the Taita Hills. We really want to kind of understand what's pollinating it, whether these are host specific, whether they're more generalist, what kind of pests go for this endangered coffee species, but also what goes for these more common species, and whether we have overlap, whether we can build an ecological network of interactions and then integrate things like chemistry and phylogeny, climate change, host range, to try and predict how and where insect pests will eat coffee, and what traits we can use in wild coffee species to integrate into domesticated species to try and help coffee production and sustainability. Again, another simple hypothesis, there'll be more overlap between wild and cultivated coffee species with respect to insect pollinator communities and for insect herbivores. So really, these are very basic, almost embarrassingly basic questions, but we just don't, it highlights the fact that we know virtually nothing about wild coffee. So we know a load about the species that we grow, and we don't know anything about the wild relatives. And this is also, this is kind of true for lots of systems. We're trying to have a colleague who works for the National Rice Institute in Thailand, and they're doing a lot of work on the wild relatives of rice, for example, what traits we can use, how we can breed more effectively for yield. The one example that we do know a lot about, something sort of fairly boring really, is brassica. So the Brassicaceae, we know quite a lot about them. A Brussels sprout cabbage family. We probably know more about brassica chemistry than we do about coffee chemistry. And I'll leave you to make a decision as to what is a more interesting and important crop out of those two. So eventually, the idea is that we want to build up. So we've got, just checking the time, we've got a proposal in progress to apply for funding to take a network approach to quantifying these communities. So what we want to do is to start really understanding climatic factors, intraspecific variation, interspecific variation. If anyone's familiar with our work on ficus, on figs, a very similar conceptual framework actually. So trying to predict when and where pests turn up, how climate affects pests, how chemistry affects pest occurrence. What traits, what coffee traits other than just caffeine, so all these other chemicals that we have in coffee, how they vary. And really the idea would be to integrate West Africa, East Africa and Madagascar to try to understand whether we have this real wet-dry gradient in herbivory and wet-dry gradient in chemical composition. Would like to build more robust phylogenies, would like to understand intraspecific variation and get at what happens on a climatic scale. And would also really ultimately like to get involved with things like the coffee berry borer, because we really only know the coffee berry borer from a couple of wild locations. Don't know much about its phylogeny, we don't know much about these wild pests as well as these wild coffee species. And it seems like a missed opportunity really to study these wild species. So ultimately we'll combine, we hope to combine insect community ecology data, genomics and chemical ecology, much in the way that we've done for other systems, try to understand how chemical and biological diversity interact. So do insect herbivores drive chemical diversity in plants? Does chemical diversity determine what eats a plant, and how is chemical diversity influenced by climate and phylogeny? So ultimately this will mean conducting much more focused experimental projects, so manipulating defensive regimes and inducing defensive reactions in coffee. And there again, so there are analogies here with Martin Wolf's work in Willow and his group's work in Willow. So we'd like to be understanding what triggers different types of chemical defense, induced defenses, constitutive defenses in coffee, and that's going to require greenhouse experiments or semi-wild experiments. So unlike willow, coffee grows it's reasonably fast, I suppose, for a woody tree species, but within about 3 years you'll get beans. So it's not ideal for greenhouses, but for semi-wild studies, plantation-based studies or studies of populations in the wild, it will make quite a nice system. So those are my conclusions. Again, as I say, very little data of our own, but a bit of a tour of the system and what we plan to do with it. So if anyone's interested, I'm of course more than happy to discuss it and have people involved for master's projects, that kind of thing. Especially others working in Africa, especially if you're happy to pick up samples of coffee plants, that would be really good. And there are a few references for those of you who are interested in these things. So this is this Adler reference is a classic. Any chemical ecologist should read that. Aaron Davis, there's a load of really nice popular science stuff on this article as well. They basically went into the rainforests in West Africa, went to a load of herbariums and kind of rediscovered this arabica-like flavor in a heat-tolerant coffee species. Yeah, this is interesting as well. The caffeine and floral nectar enhances a pollinator's memory of reward. So it's a bit of insect behavior there, and this caffeine really has multiple functions. So I hope, I haven't probably done it justice, but I hope that gives some idea of what the kinds of things we're wanting to do, the kinds of things we're doing. And the main reason for giving the talk really is just a bit of an advert, something to think about, especially if you've got African focused research and you're interested in collecting these extra data sets or collaborating. So I've got, I think, plenty of time for questions, because that was a bit quicker than I expected. That was only a 20-minute presentation. So I've got some time for questions if anyone has any.
Okay, so anybody can just start speaking directly. I can start with my question. What is do you have any idea? You know, the coffee is of course spread throughout the tropics.
That's something that